Good afternoon, everybody. My name is Amer Mohamed. I'm a Cancer Therapeutics Fellow at the Department of Cellular and Molecular Medicine, School of Medicine. My presentation today under the title of A Signature of Autocrine Autonomy in Cancer Cells. Today, I'm going to show you how we transfer from a basic science research into a clinical research. As we know, most of the studies exploring gene expression data result in one or more gene signature. What is the gene signature is? Gene signature could be a single gene or a list of genes sharing a common pattern of expression that can be used to classify groups of samples, groups of people, in any independent data sets. The scientific community and the clinician have been used this strategy to understand the tumor biology, to prognose, to predict, to diagnose, and to predict the treatment of many diseases, including cancer. Among cancers, breast cancer leads the way with the most FDA-approved signatures, and the first gene signature approved for the breast cancer was the Mohamed, which consists of 70 genes and can prognose the estrogen positive, HER2 negative, with lymph node negative and positive patients. And then other two signatures have been approved by the FDA, Oncotype DX21 genes and BAM5050 genes, and both are using for the prognosis of the estrogen positive, HER2 negative. Another two gene signatures are not approved yet, but doing well for the prognosis of the estrogen positive and HER2 negative. As you can see here, the gene signatures, which approved and also one that not approved, doing well with the prognosis of the estrogen positive and HER2 negative breast cancer submolecular types. However, there is some limitation that not addressed yet for these gene signatures. Among these limitations, the ability of these gene signatures in predicting relapse may be dropped after the first 5 years from the diagnosis, and also the applicability with respect to targeted therapy is still primarily limited, and also none of the gene signatures can be used for CTC datasets, circulating tumor cells, which is important for the metastasis. And also, most of these gene signatures are not universal. You can see that one of them can be used in the breast cancer, but you cannot use it for the colon cancer or prostate cancer. The main cause of this limitation is the lacking of a sensible biological meaning of these signatures. And fortunately, I'm working in a group that works in something related to the cancer cell, which is one of the hallmark of cancer, which is the ability of the cancer cell for self-sufficiency in the growth factors, which in another meaning you can say that autonomous cell growth of the cancer cell. What does it mean, autonomous growth of the cancer cell? The autonomous, as a word, it comes from the Greek roots, auto meaning self, and nomos meaning the law. That means that the cell have the ability to grow and independent on the external factors, growth factors I mean, and to explain the autonomous growth of the cancer cells, we know that if you have a normal cell and you see that in a media, you must put growth factors in the media, otherwise the normal cell cannot grow. But if you have a cancer cell and you didn't put a growth factor in the media, this cell can still grow. To explain what is the autonomous cancer cell survival and replication, scientists put a lot of mechanisms. One of them is the autocrine secretion or the paracrine secretion. The cancer cell itself secretes something like growth factors, and then or growth factors like the platelet-derived growth factors, transforming growth factors, as well as the epidermal growth factors. And then the cancer cell has the ability to send this growth factor and take it again to survive. And the paracrine secretion, the cancer cell itself sends the growth factors that is secreted by the neighbor cells. Another mechanism that explains the autonomous cancer cell growth is the abnormalities in the growth factor receptors. There is upregulation in the growth factor receptors happen to in the cancer cell. For example, in the squamous cell carcinoma, as well as in breast cancer, they notice that there is upregulation in the epidermal growth factors. Also, the poor fidelity during the DNA replication, as well as the abnormal signal transduction, contributed to the autonomous cancer cell growth. Among these four, serum-free cell culture studies implicated the autocrine secretion of growth factors as key support for intracellular mechanisms that impart autonomy. And fortunately, I was working in a group that discovered an eukaryotic circuit for autocrine autonomy. This circuit imparted the cell's ability to survive in the absence of the growth factors, and this circuit assembled when the monomeric GTPase get coupled with the heterotrimeric GTPase in the presence of a linker protein, and this linker protein is a metastasis-related protein in different solid tumors and named as GIV or Girdin. And once the linker is connected the mono-GTPases with the trimeric GTPase, it leads to a closed loop circuit, a closed loop control, which make the mono-GTPases and the trimeric GTPases correlate each other, and then lead to the sense and the secrete circuit to work. So our question at this point was, what happens if I'm going to make knockout for the linker protein, which is connected the mono-GTPases with the trimeric GTPases? So to answer this question, we did a protein-protein interaction map specific for the Golgi, and we found that if we deleted the linker protein, which is GIV at this point, it will affect the sensing proteins, which is in the yellow color, and the secretion proteins, brown colors, as well as the survival proteins, which is faint green color. To confirm this protein-protein interaction network map, I did two experiments. The first one is in a 2D model, and second one in the 3D model. In the 2D model, I take the cell line, one with GIV, which is the parental one, which is autonomous circuit is working, and the one without the GIV, the autonomous is disabled at this one. Then I seeded these cell lines in different concentration of the FBS. I will start with the 10% FBS. You can see that there is no difference between the one with the autonomous working and the one with the autonomous doesn't work, because you have, at this time, you have the growth factor in the media. But if you look at the 0% FBS, you haven't the growth factor, you will see that the coupled one or the parental one, which had the GIV and they have the circuit working, has the ability to survive. But the one without the GIV, where the circuit is disabled, you will find that the cells is died. So I did flow cytometry to determine which type of this will happen, and I found it is apoptosis and necrosis as appear in the figure. And also in the 3D model for breast cancer cells, you can see that when the autocrine is abled in the presence of the linker protein, which is GIV, you will see survival for the tumor sphere. But in case of the knockout of the GIV, which is the linker protein, you will find the cells were not able to live and it dies. So the coupling of the mono-GTPases and the tri-GTPases by the linker protein GIV generates secret and sense autonomy to work, lead to the survival. However, if you take it out, it leads to the death of the cell. So the question of the clinical at this point is arising. And we asked what is the genes or the gene composition of this autocrine autonomy. So I take the cell line system, one with the autonomy enabled and one with the autonomy disabled, and I seed it in 0% FBS to confirm there is no growth factor there. And then I extracted the RNA, I did RNA sequencing, and then I did differential expression gene analysis. And I got 29 genes upregulated, and the 3 genes are downregulated. And we named this gene signature as the autocrine autonomy signature or Girdin print. Then I did reactome analysis for the up and the down gene list of the Autocrine autonomy signature, and I found that most of the gene signature which involved in the gene signature are involved with the growth factor signaling like EGFR and others. Then I tested the expression of the gene signatures in the different types of the breast cancer. As we know, the breast cancer has different types: luminal A, luminal B, HER2 positive, and the triple negative. And I found that my Girdin brand is progressively increased from the normal to luminal A, luminal B to HER2 positive. And also I compared my gene signature with the other FDA-approved gene signature, and I found that my gene signature is upregulated in the different types of the submolecular types of the breast cancer. So the question at this point is, as the gene signature is arising from autocrine autonomous cell growth, so probably it will affect the metastasis. So I tested my gene signature in comparison with the other FDA-approved gene signatures in terms of calculating the metastasis-free survival. So I found the Mohamed, Oncotype DX, PAM50, all of them are FDA-approved gene signatures, but cannot prognose the metastasis-free survival after more than 10 years. But my signature did that with significant p-value. As the both cohort have different cycles, like lung metastasis, bone metastasis, brain metastasis, so I tested my gene signature in comparison with the FDA-approved gene signature for the different cycles. I started with the lung metastasis, and you can see here, all of the gene signatures, which is FDA-approved, did well. However, my Girdin brand were superior to the FDA-approved signatures. And also I tested for the brain metastasis, and also I found that Oncotype DX failed to detect the metastasis-free survival. However, the PAM50 and the brain signature, who is from Masagi group, which published in Nature, and the Mohamed brand did well, but my Girdin brand was superior to this gene signature. And also I tested for the bone metastasis, and I found that my gene signature, which is the Girdin brand, are superior to other gene signatures. Then I tested my Girdin brand in the lymph node negative breast cancer patients, and I found that my Girdin brand can separate the high and low risk groups in these patients too, with p-value significant. As I said, no other signature can be used for detection in the CTC, the circulating tumor cell, which is important for the metastasis. So I tested my Girdin brand, and I found it can use for the CTC dataset in comparison with Mashaurin group, which used for the CTC score, and mine was significant in the separation of the low and the high risk people. Also, I tested my Girdin brand in different types of cancer, like leukemia, urethral carcinoma, colon cancer, lung cancer, and neuroblastoma. And I found the Girdin brand did well with B significant value, and it can separate the low and the high risk group of people.
How do you define high and low for in each case?
We calculated based on a step minor threshold on the composite score of the gene separation value.
So how is that threshold decided on?
The threshold is decided upon a step minor function, calculating the step function.
Okay, so how do you explain that in some of these situations that you looked at, the groups are not balanced in terms of low and high? So, for example, for colon cancer, you have only, I don't know, 20% of people are in the high group.
We just calculated the total expression of these genes in this group and gave them a composite score. So the conclusion of this study, it seems that Girdin brand is superior to other signature used in the breast cancer prognosis. In-depth analysis showed that we can use this gene signature in the CTC. And the Girdin print works well in different types of cancer. So we are aim to make patent for the gene signature, but we still need to make validation in different data sets. In the end, I'd like to thank my primary mentor, Dr. Ghosh, and the help of Dr. Saho and Dr. Saptrashi. This work was funded by the NIH-funded Cancer Therapeutics Training Program. Thank you so much, and welcome to any questions.